Hello everyone, and welcome to today's webinar titled Introduction to the Water Quality Simulation Program, known as WASP. This webinar is sponsored by EPA's Water Modeling Workgroup, and my name is Jason Gilday. I'm with EPA, and I'll be moderating today's webinar. And thank you for joining us today. First, I'm going to briefly summarize the webinar logistics as soon as I can get my slide to advance. There we go. We hope everyone will have a lot of questions today. The phone lines will not be opened, but you can type your questions into the questions box on your control panel and then click send. If your control panel is not showing, simply click on the small orange box with the white arrow to expand it. If you have any problems viewing the broadcast, just let us know in the same questions box and we will post an answer in that same box. A phone number is also available on your control panel if you are having trouble hearing audio through your computer speakers. This webinar will be archived for later viewing on EPA's Water Modeling Workgroup website, and we will provide that archive link for you on a slide at the end of today's webinar. Also, at the end of the webinar today, you will be able to download a certificate of attendance for participating, and we'll provide directions for downloading it at that time. Lastly, the materials in this webinar have been reviewed by EPA staff for technical accuracy. However, the views of the speakers are their own and do not necessarily reflect those of EPA. Mention of commercial enterprises, products, or publications does not mean that EPA endorses them. Now that I've covered the logistics, I'd like to provide you to a quick introduction to this water quality webinar series. The purpose of the series that we've been putting on is to help water quality professionals better understand water quality modeling and how models can be used to solve the problems facing water quality regulators. Today's webinar is the 13th in our webinar series. All of the past webinars have been recorded, archived, and posted to EPA's YouTube channel website. Links to the archive files are available on our Modeling Workgroup website, and you'll see the link at the bottom of the slide. And in a few weeks, an archive of today's webinar will be posted on the same website. So for the audience, these webinars are targeted at water quality professionals who may use, want to use, or review models as part of their job, but who do not necessarily have a modeling background. As such, we're assuming you have basic understanding of water quality science and concepts, but perhaps have limited direct modeling experience. So with that, I think I have all the logistics out of the way. I'd like to introduce our speaker today. Today's speaker is Tim Wool, and Tim is a national TMDL expert with the US EPA from Region 4, that's in Atlanta, Georgia. And Tim has over 25 years of experience in the development and application of water quality models that are used for TMDL development and waste load allocations. Tim established the Watershed and Water Quality Modeling Technical Support Center located at EPA's Office of Research and Development Laboratory in Athens, Georgia. At the center, he was responsible for the development and establishment of the TMDL modeling toolbox, which brings together a collection of models and tools to streamline TMDL development. Tim is one of the primary developers of the Water Quality Analysis Simulation Program, WASP, which is routinely used in the United States and throughout the world. And Tim has supported EPA with developing a technical approach for numeric nutrient criteria for the state of Florida, and he has significant experience in developing and reviewing TMDLs for bacteria, nutrients, metals, dissolved oxygen, and mercury. One last reminder, we'll have 2 question and answer sessions today, but we often receive more questions than we can actually answer. We'll do our best to get through as many as we can. We do have a bit of a long webinar today, so we might have to cut some of the question and answer sessions a little short. Throughout the presentation, though, please do type in your questions as you think of them, and we'll get to them periodically throughout the day. One more thing, today's webinar is, we're trying something a little different. The webinar today has actually been pre-recorded, but there will be a live question and answer session. So we'll be pausing periodically and for question and answers, and Tim will be on the line to answer those. And with that, I'm going to pass it off to Tim to start the webinar. Thanks.
I want to thank everybody for coming to the webinar today. We found these webinar series to be very useful and well supported by the modeling community. Today I'm going to give you an overview of the Water Quality Analysis Simulation Program, WASP-8. My objectives are to give you a brief history of WASP, a description of how the WASP modeling framework works, talk a little bit about how different water body types can be simulated with WASP, an overview of the data needs of WASP and where you can potentially get that data. I'm going to demonstrate several utility programs that are readily available for use with WASP. I'll give you an introduction to the WASP interface where I'll actually build a data set, and then in the end we'll look at how we can review model results and talk about the calibration procedure. I'd like to give you a little bit of history about WASP. It's been around for a long time. It was originally developed in the 1970s by Hydroscience, which later became Hydroqual and now is HDR, to investigate eutrophication issues with Lake Erie. In 1983, it was brought to EPA by Bob Ambrose at EPA's ORD laboratory in Athens, Georgia, to investigate water quality issues associated with the Delaware. In 1985, the mini mainframe version of WASP was ported to the personal computer environment, which also was significant because this was the first time that the model was available to the general public. In 1994, the graphical user interface and post-processor for Windows was developed by ASCI Corporation. This is a company that James Martin and myself worked for at the time. ASCI was gracious enough to donate this version to EPA for general distribution. In 1999, we developed the hydrodynamic linkage application program interface, which allowed WASP to communicate with three-dimensional hydrodynamic models such as EFDC. In 2001, WASP-7 was released and it was geared to supporting the TMDL program. So a lot of the enhancements that were made in the earlier versions of WASP-7 were directly related to supporting decisions that had to be made in the TMDL program. From 2001 to current, there's been numerous upgrades to the model. The addition of multiple algal classes, multiple BOD classes, the inclusion of benthic periphyton algae, the sediment diagenesis routines, pH alkalinity, and then also the ability to get data from different tools such as databases, and then to make post-processing easier to do. In 2016, WASP-8 was released, which was a major update to the model because much of the source code and interface was redesigned. In WASP-8, there was a rewrite of the user interface, or what I'm going to call a modernization of the interface. The previous version of WASP that was developed in 1994 by ASCI Corporation used a set of software development tools that are no longer supported and available for the Windows environment. Thus, it was only a matter of time before WASP-7 would not work in modern Windows frameworks. So the new interface was developed using an open source environment. So all of the tools that were used to develop, to compile and develop the interface were available freely. We improved linkages to external databases to make it efficient to bring in large amounts of time series data into the model. The interface was improved efficiency-wise, and we learned from our mistakes in the past to make it a little bit easier to use for the user, but also to allow it to be used more efficiently in a batch mode. The models themselves were rewritten using more modern Fortran algorithms. All of the memory is dynamically allocated, so really there is no limitation to the number of state variables or segments or time periods that you can simulate. It's all a function of how much memory you have on your computer. We gave greater control to the user for picking which state variables they would like to use in the model, because what you'll see in a little while, WASP does not make you use all of them. You can use what you want. We improved the runtime performance. WASP-8 is probably about 2.5 to 3 times faster than WASP-7. And we've spent significant amount of time to improve post-processing performance because as we move forward in water quality modeling, our water bodies that we take on have become more and more complex, larger systems. Thus, the model output becomes larger in size, and to render it and look at results more quickly was an important aspect that needed to be considered. WASP 8 is now available in both the 32-bit and 64-bit distribution. The 32-bit version is only specific to the Windows environment. The Apple and the Linux version are available only in 64-bit. So the decision was made during the rewrite to go ahead and target multiple operating systems to make WASP more widely available. So we have now supporting both Windows, Apple, Macintosh, OS X, and Linux. Next, we're just going to talk about what is WASP. WASP is one of the most widely used water quality models in the world. It has a long track history of being applied to rivers, lakes, streams, estuaries, and reservoirs throughout the United States and the world. It is a mechanistic model in that it uses scientifically based algorithms to relate water quality concentrations to response variables in the environment. It is a time variable continuous simulation model, which means it basically can simulate water quality through time under different environmental conditions such as meteorological conditions or loading conditions. It's capable of simulating both conventional pollutants and chemicals. There's two modules that are available with WASP. The first one is the conventional pollutant model, the eutrophication model, which does the nutrient cycle, algae, periphyte, macroalgae, dissolved oxygen, so on and so forth. And then there is the chemical model, which is capable of doing organic chemicals, nanochemicals, and nanosolids. Just a little bit about the utility of a mechanistic model. We always have to realize that models are a simplistic representation of reality. While it is a mechanistic model, it cannot simulate everything. Therefore, all models are wrong. But hopefully we can find it useful. Now, it does have the ability to interpolate between known and unknown conditions, and it's because of its mechanistic basis. It provides a linkage between loads and response variables. It can determine important processes within the water body. It can be used to determine load reductions to meet water quality standards, whether they are never to exceed or never to exceed X% of the time, or that the standard is expressed as a duration, frequency, and magnitude. And then it's also can be used to evaluate best management practices. WASP can be applied both as a time variable or a steady state model. In a steady state model, you would just set all of your time variable conditions to a constant value and let the model iterate to a steady state example. Steady state approaches typically used for criteria development and waste load allocations. For nutrients, it's typically not a steady state condition. Usually it's not a specific critical condition. There is seasonal variation, and the things that need to be considered are varying meteorological conditions such as low, average, high flow years. In which order do they occur? So the long-term simulations allow us to evaluate over the range of environmental conditions. So when it comes to mechanistic models, here are the kind of the pros and cons. The pros are you have a direct linkage between stressors and response variables. In our case here, if we're looking at nutrients, nutrient loadings, we can determine their effect on chlorophyll, whether it's algae, benthic algae, or macrophytes. We can also look at the effect of nutrient loadings on light availability and light reach and bottoms for seagrass propagation, or impacts on dissolved oxygen. They are also useful for extrapolating amongst environmental conditions, so changing in environmental variables like light, flow, or we can also look at compare it between extrapolate between a current condition and a water quality standard condition. We can determine how long it would take for the water body to respond to management practices to meet a goal. and we also can evaluate duration and frequency of water quality conditions. Some of the cons are it's time consuming. They do take time to set up these models and gather the data. It could be costly to collect the data that you would need to adequately calibrate the model, and certainly they can be misapplied. I'd like to provide you a little background on the WASP framework and how the different components interact. WASP is not like most of the other models in that it is an integrated package where which allows us more flexibility to have different model types. When we look at this, the thing that you run called WASP is actually the user interface, and it reads a binary input file that can only be read by the interface. But once that information is into the interface and it's set for a specific model type, when you hit run, it actually will load a dynamic link library, which is the specific model. So if you're developing a eutrophication model, all of your input in the preprocessor will be configured for the eutrophication model. And once it starts to run, information is passed from the interface to the module. No files are actually read at this point. Everything is facilitated by dynamic link libraries. If you're linked to a hydrodynamic interface file, that information too is served to the model via dynamic link libraries. When the model is running, it writes information out to an output file, which is a BMD file. Again, this is another binary file that has been optimized for rendering. That can be read by the Water Resources Database graphing program, and there's also utilities available for extracting information from the BMD file to take to spreadsheets and so on and so forth. This framework allows us to develop different model types without actually rewriting or reconfiguring the interface itself. The interface gets all of its information from a database file, so that if we create a new model type, we would just modify the database and that model type would show up. The interface was also modified to allow for access to data, time series data from different sources such as databases, spreadsheets, and ASCII flat files. The WASP structure really has not changed since it was originally developed in the 1970s by Hydrosciences. There's two main blocks of code or modules. The first one is the transport and solution technique module, which controls all of the calculations for water movement, solids movement. And then the bookkeeping part is the one that does the mass balance calculations as well as the solution technique. And this approach allows us to develop several different kinetic modules that interact with the transport bookkeeping block that are specific to water quality conditions that we want to simulate. So in the case of the advanced eutrophication model, the yellow box would contain all of the algorithms that would have all of the transformations and calculations associated with conventional water pollutants. In the case of the organic chemical model, they would have the specific algorithms associated with chemical interactions in the aquatic environment. And what we do is we write a specific yellow block for each model type, and it gets linked into the transport block, and that creates the individual models, the organic chemical model and the eutrophication model. This is quite handy because when you look at it, you have a separation between transport and kinetics that you can look at how the each one affects water quality. WASP is considered a box model in that the water body is not to assume to be some structured grid where you have number of cells in one direction and number of cells in another direction. The model does not assume any specific conductivity. It's up to the user to define the conductivity, or if you are linked to a hydrodynamic model, that information is provided within the linkage file. So we break the water body into boxes. Boxes are kind of a way to show it, but just imagine it is blobs of water that interact with one another. And each one of these boxes actually makes up a computational element where all the state variables are recalculated for each box at every time step. The nice thing about a box model is it allows the most flexibility you could have when it comes to breaking your water body up. It could be done in 1 dimensions, 2 dimensions, or 3 dimensions, or a hybrid where you have one-dimensional going into two-dimensional going into three-dimensional. So it allows the most flexibility. And we'll see some examples of this in the transport section. It also allows us to control spatial complexity in that we can model our water body as a single continuous stirred reactor, or we could add more segments to resolve different gradients or different management questions. So it gives us a good handle on how we can control how we break our water body up. Okay, the water quality modules that are available with WASP-8 consist of the advanced eutrophication model, which simulates dissolved oxygen, up to 5 biochemical oxygen demand state variables, the nutrient cycle considering nitrogen, phosphorus, and silica, 5 phytoplankton species, 5 periphyte and macroalgae species, 10 solids groups, 5 bacteria group, water temperature, sediment diagenesis, pH, and alkalinity. So all of those state variables are available in the advanced eutrophication model. In the toxicant model, you have 10 chemicals, 10 nanochemicals, 10 solids, 10 nanosolids, DOC, kinetic sorption, partitioning, degradation via photolysis, volatilization, ionization, hydrolysis, so on and so forth. And then you also have reaction yields or paths where one chemical can degrade to form another. Another module that's currently under development and should be available sometime next year is a metal speciation model, which will calculate or predict metal species as a function of EH, pH, and complexing components that are available. So we'll talk a little bit about the advantages of WASP and some of its features. By far, one of the biggest advantages of WASP is its network flexibility. It can be virtually applied to almost any water body type at any level of complexity. It considers most of the water quality problems that we're faced with, conventional water quality and organic, chemical, toxic fate. It allows us to look at the separations of processes on water quality conditions, whether it's transport or kinetics. The modeling framework provides extensive linkages to other models and spreadsheets and databases. The model also has 3 different solution techniques available to the user. The most widely one is the Euler solution technique. When using hydrodynamic linkages, you have the cosmic, which is a flux limiting one, which kind of controls numerical dispersion. And then you have an intermediate predictor corrector Runge-Kutta solution technique. So those are some of the different advantages. There's a lot in this framework that allows you to do your water body. Some limitations, which are kind of critical. One of the things is WASP does not do mixing zones. It assumes once something comes into one of the boxes or the segments, it's completely mixed. So it's not going to be able to do things like core mix. It cannot handle non-aqueous phase liquids like oils because they could be either on the bottom or the top of the water column. The model would assume that they're completely mixed. And we talked about the metal speciation, which is not available yet. But one of the big advantages of WASP is the ability to control variable complexity. And when I say variable complexity, it could be a couple different things. It could be how the water body is divided up. It could be how water quality state variables are considered in the model. We already talked a little bit about segmentation. You can have as many segments or as little segments as you need to answer your question. But you also have the ability to control which state variables be used in the model. So in essence, really, WASP is not a model until the user starts adding the state variables in it. WASP is actually a systems of equations that have the built-in relationships amongst those different state variables. So if you wanted to do a simple Streeter-Phelps calculation in WASP, the only thing you would need to put information in for is DO, BOD, and water temperature, and the model would do all the calculations. A lot of the other water quality models that are out there you have to do all or nothing, which basically means you have to simulate all of their state variables, provide all of the information for each one of those state variables before we would actually make an appropriate calculation. So things that are going to control the complexity is the aquatic system, rivers and streams, depending upon what kind of gradients that you want to preserve longitudinally, will determine how many segments you have. For lakes and reservoirs, what kind of gradients do you have as far as top to bottom for water temperature, dissolved oxygen, and laterally as a function of water quality. And then for estuaries, where you have different gradients caused by freshwater flow and salinity, how much of that do you want to make sure that you capture the gradients? You would also adjust the complexity of WASP based upon the chemical behavior, whether it's I need to do Streeter Phelps, or I need to include the impacts of phytoplankton and sediment fluxes in our calculations. Or it could be the behavior of specific chemicals in the environment that degrade to form other daughter products that need to be accounted for. And then, of course, the model complexity can be changed to address the management question. You know, I always use in the WASP class, if you wanted to know how much phosphorus is in Lake Superior, how many segments would you need? And the answer is actually 1, because that would tell you how much is in Lake Superior. But if the next question is you wanted to know where it was at, you would have to redefine your segments to better account for where the phosphorus is in the system. So again, one of the biggest strengths of WASP is it's a variable complexity and how you apply it, and you can adjust as you apply the model. All right, let me just give you a brief overview of how transport and flow is considered in WASP. We talked about segmentation, and as I told you, WASP can be applied in 1, 2, or 3 dimensions. In our case here, the top one is a continuously stirred reactor, where you just have mixing within that one segment, where you can have an inflow and an outflow. And down at the bottom, you can have a set of segments in series, mimicking a river, where you have flow in and flow out. Not only does WASP consider the impacts of water quality in the water column, but for each one of your segments, you can actually have interactions with the underlying sediments, whether they're for sediment oxygen demand, nutrient fluxes from the sediment, or the interactions of organic chemicals with solids and settling out. So we can do that. You also can apply the model in 2 dimensions, where in our upper box we actually have stratification between the upper and lower waters, where you can have exchanges and flow between them. Or in the case of a stream network, you can have a river that breaks off into branches and comes back together, or you can have tributaries coming into your segment. And you'll see a little bit more of that when I demonstrate the WASP network tool. And again, here you can have the sediment segments as well. For most cases, if you're going to apply it in a 3-dimensional mode, you're probably going to want to link it to a hydrodynamic model. In the example here, this is the Neuse River estuary that I applied WASP to several years ago. There's 405 horizontal cells for 4 layers. And if you were to do this yourself, you would have to describe to WASP the transport between each one of those segments for each one of those layers. So you would more than likely apply a 3-dimensional hydrodynamic model and link it to WASP. So our options for moving water are, there are some, the first 2 are simple flow options, a net flow, where you basically, as a user, would specify the flow coming from one segment and going into another. So it would be flows across the boundary to a segment, and the flow from that segment to the next, and the model would calculate what mass gets moved with it. Or you can have separate flows that would basically circulate water, where you could have flow from one direction in a segment and flow from the opposite direction, and it would move mass in both directions. For stream network flows, you have several different options that you can specify how the model would calculate flows. The first one, and probably the most prevalent, would be the calculation of a kinematic wave for free-flowing reaches, which would take into account segment geometry and slope to calculate a water wave down the network as a change in flows. You also can have ponded reaches where you can have a weir and set a weir height, and it would calculate the overflow over the top of the weir. You also can have dynamic flow where you have backwater. So flow is not just a function of slope, but it's a function of water surface slope or water surface elevation. So you can actually have backwater effects, tidal effects that the model will calculate. You also have the ability to have internal transfers of flows where you can transfer water from one segment to another by using a time series. It could be used to mimic dam releases or water withdrawals. And the last one is you can get the calculated flows from an external hydrodynamic model. So this just to give you an idea, and one of the big strengths of WASP is your whole network does not have to be one of the flow options. It can be a combination of all of the flow options except the hydrodynamic linkage. If it's hydrodynamic linkage, it has to be doing all of the transport. So in our case here, we have segments where we have free-flowing main segments where we could be using kinematic wave or the descriptive flows to calculate the flow. In our case here, we have a ponded segment that has a berm that once water comes up here and it gets higher than the berm, it will come into this segment. We have the free-flowing segments that are coming into a segment that's just upstream of a lake or a pond. And in this segment here, we would calculate a backwater effect as a function of the water surface elevation of the pond versus the flows coming in. And then we have a dam release or a weir that allows water to flow downstream. So you can link all of these different flow options together in a single network. I wanted to just give you a brief overview of the sediment transport options that are available in WASP. There is a fully mechanistic based sediment transport model in there that's a function of shear stress on the bed, particle size, and density. You could parameterize up to 10 different solids based on density and solid size, particulate sizes, and it'll calculate both settling and resuspension as a function of changes in flow and water velocity. Or the old method that's been in WASP for years is the user has the ability to define settling and resuspension rates both spatially and temporally by using segment-specific rates as well as time functions. Sediment transport definitely is an important aspect that needs to be considered when doing organic chemical modeling. Alright, we're going to look at some of the data requirements, what kind of data is needed for WASP and where you can get it. So really the types of data that you need are kind of broken down into a couple of different things. The first one is the model domain, the how you break your water body up into segments. And that's things like, how do you define the channels and such. And we'll talk a little bit more about that in depth in a little bit. And then one of the most important components you have to consider when you're applying the model is where are you going to get boundary data for the model. So a boundary is where you have information coming from outside the model network into the model network. We are getting better at simulating larger areas, and if you're simulating whole watersheds, that's great, but you still have to figure out where you're going to get the flows, the water quality concentrations of the constituents that you're simulating at, everywhere where there's a pour point where flow and mass is entering the water body, or in the case of flow for downstream where you have dynamic flow, where you're going to get water surface elevation data. The other types of data that you're going to need is environmental conditions data, and this is things like solar radiation, air temperature, and a host of information that you would use to calculate water temperature, as well as provide environmental conditions for calculating light and available light for phytoplankton growth. Some other things that you might have to consider, whether it's atmospheric deposition for conventional pollutants, where you can provide a time series of nitrogen and phosphorus and BOD loadings from the atmosphere, or if in the case of it's an organic chemical or a chemical in general at the atmospheric deposition rate for that. So the first data type we'll talk about is the model network. For streams and rivers, we have the National Hydrography Data Set, NHD+, which has a lot of information that has been both interpolated from satellite imagery and then field verified. But what we get from NHD+ is we get lengths, widths, depths of the stream or river network under average flow conditions. They give us the bottom slope, the average flow, and the cumulative drainage area upstream. And we can get the information from the NHD Plus coverage, which you can get as a GIS coverage, put it into a spreadsheet and develop our segmentation and then paste it into WASP. And I'll also show you another method where you actually, there's a plug-in tool with basins that allows you to develop a WASP network from NHD Plus. But where do you get this information? Well, there's a website for downloading the NHD Plus information, or you can download it directly inside basins and then process it with the WASP network tool, which I'll show you in just a little bit. The other way that you can get stream and river network is from topographical maps or individual stream surveys. There's not as much detail in these topographical maps as it's in NHD+, so you're much better off going there for that information. For lakes and reservoirs, again, you're going to be counting on topographical maps, bathymetry maps from the Corps of Engineers, if it's a Corps of Engineers managed reservoir, or Tennessee Valley Authority, or whoever is managing it. Or you could go to power utilities. They have maps and coverages that's typically found with their FERC licensing when they need to go through that. So you can go to those individual entities to get both topographic and bathymetric maps of your lakes and reservoirs. For estuaries, you're typically going to go to the NOAA nautical maps or coverages, and they have both spatial and depth components associated with them. Next, I'm going to give you an overview of a utility that's available as a plug-in to EPA's Basins Data Model Frameworking Tool, which is basically a public domain GIS system that has many utilities that are good for developing models, analyzing data. So I'll just give you a quick overview of the WASP network tool. I'll give you a little demonstration. It is a plug-in. As far as what kind of data the network tool needs, you'll need the National Hydrography Data Set, NHD+. You could download USGS streamflow information, not only gauge location, but the time series of data to pull directly into WASP. And then if you built a water resources database project that actually had quality or concentration data, you could pull it in at the same time. The nice thing about the Basins framework is it can download the NHD Plus network for you, as well as the US streamflow information from their NWIS system. So what it's going to do is it's actually going to build a WASP network based upon the characteristics you select from the stream network. And this not only includes doing the main stem, but it'll do tributaries, as you'll see in a moment in the demonstration. And it'll apply simple rules for segmenting the model, the network, into segments that are appropriate for the model. It's not exactly perfect, as you see, but you have ways to manipulate and smooth your network out. It'll take care of generating all of the flow paths, so where water comes into the WASP network and then where it goes from there. It works for all WASP model types, and then we'll import it directly into the WASP model. Okay, what we're going to do is we're going to load Basins. We're going to say build a new project, and what Basins does is it brings you up a map of the United States and all of the HUC-8s, and what it allows us to do is select a specific HUC-8. So I'm going to use the zoom key. I'm going to zoom into Georgia. I'm going to find a HUC-8, the one I'm hovering over right here, which is Briar Creek. I'm going to use the select tool to select this HUC, and you'll notice that it gets highlighted yellow, and then over on the left it says Briar, and we're going to build a project. Now, I typically like to create my own directories with the watershed name and not use the HUG number because I forget what the HUG number is. I'll create a directory, and then my project name I will give it with the watershed name here, Briar Creek. And once this is done, I'm going to click OK. It's going to let me set the projection. I'm just going to use UTM 17 for this area of Georgia. And basins will go off to a server and it'll download some base coverages for this Briar Creek watershed. We have things like permit compliance system, permits. I wouldn't trust this one because this data is kind of old, but it's pre-packaged. So that just gives you an idea of some of the basic data that you would get with creating a project in basins. Now what I'm going to do is I'm actually going to use the data download tool, and I'm going to download specific data. But from here we can get NHD Plus data, USGS NWIS data, STORET data, National Land Cover data sets. You can just download all that. It comes into your projects. I'm going to select the NHD Plus catchments and the NHD flow lines. And I'll end up with a project that looks like this. So this actually has all of the channel geometry for all of the stream segments in the system. I'm going to shut off a couple of these coverages. I don't need the area features or the catchments. And ultimately, the only thing I'm really interested in is the flow lines, the hydrography. I'm going to change the colors here to make it a little bit easier for you to see. So that once we use the WASP network tool to create our segmentation, you'll see a little bit better. So we're going to ask it to develop a WASP network for this stream or this watershed. So we're going to go to the models, click on WASP, and then what we need to do now is we need to go down and select the bottom lowest point of the stream that you want the WASP network for. So in this case we're going all the way down to the bottom. We're going to select that bottom arc, and what the tool will do is generate a network from there upwards in the network based upon the criteria. In our case here we're going to use all the way up to the third order streams. We're going to tell it to generate that, and what it's done is it's transversed the network and it's come up with the segmentation for this system. Now the initial segmentation is going to be kind of rough. It is going to take a little bit of work to clean up, but the WASP network tool provides all of the tools that you need to do that. You'll see that we have large segments next to small ones. Our goal here is to make the travel time or the lengths of the segments approximately the same to lower numerical dispersion. So the first thing that you might want to try is up at the top, you can ask it to regenerate the network based upon travel times, and it'll regenerate it. And you might have to do this a couple of times ultimately until you get what you want. I'm going to do it one more time here just to reduce the number of segments because that's probably just a little bit too many. And again, I'll just do it again. And then I'm sort of happy with what's left here. What this field here lets me do, it actually lets me have the WASP segment numbers show up on the GIS map. I'm just going to zoom into an area here to show you some issues that we might have to deal with. We may not want to have segments for these smaller tributaries. So what I'm going to do is I'm going to select one of these segments and highlight it, and then I'm going to go back to the network tool and I am going to ask it to delete it. And what the tool does is it'll highlights it in the table, deletes it, and then it renumbers and rebuilds the whole network again. And you could go on and remove the ones that you don't want to consider as you go forward. And this is an iterative process. You take your time because once you get a model network set up correctly, it's better. Now in this case here, I'm going to select several segments and I want to combine them into one because they're either too small or I just don't need spatial resolution there. So you'll notice in the table they were selected. I hit combine and then the two becomes one and the whole network is regenerated again. The same thing is true for splitting. So if you had some segments that were too long and you wanted to get more spatial resolution, you would select that segment, go to the network tool, instead of combine or delete, you would use the combine the divide option. And the divide option will pretty much, in this case here, split it into two. So now we have, instead of in that one area one segment, we have two. So that kind of gets us the functionality to that. And would be an iterative process until you get a model network that you are getting the gradients and such that you want. Once you have your network complete, you actually click a button and it'll create a GIS shapefile of this network, which is useful for publications as well as animating results. And what we're doing now is we're asking it to build the WASP input file. And what it's done is it's created an ASCII file that will import into the WASP model. You can have a look at this file. It's actually stored in your My Documents WASP folder to see how the information is laid out there. And basically the next thing we'll do is we'll load WASP 8 and we'll import this into WASP. So with WASP 8 loaded, we will go to File, Import, and from here we'll navigate to My Documents directory, the WASP folder, and we're going to look for an INP file with our project name on it, which is Briar Creek. We're going to do a import all and then set unique segment names, which brings all of that information into the interface. So what we've gotten from here is we have all of our segment information, names taken from the NHD+, but more importantly, we have all of our channel geometry, and this channel geometry is at average flow, and then we have all of our segment conductivities and where we would have time variable flow information that we would enter later. So this tool is very valuable for developing WASP networks very quickly. It doesn't really matter the size. You could do the whole Mississippi Basin, or you can just do a segment of a river network. Okay, the next set of data that we're going to talk about is boundary data. And boundary data is wherever you have flows coming from outside the model network in. It could be the headwaters of a stream. It could be a wastewater treatment plant. It could be a tributary, or it actually in WASP, boundaries can also be where water is leaving a system, because in the case of a reversing flow you'd have to put concentrations on it. So if you look at the figure on the right, this is the example that we'll be running a little later on. This is a stream model that has one main stem and 3 tributaries. Everywhere there's a star there, that's where a boundary is. So for the boundaries, I'm going to be responsible for defining the flow and the concentrations coming in with that flow for all of the state variables that I am simulating. If you don't provide a concentration for one of the state variables, it's going to have 0 concentration coming in at that point. So it's really critical to be able to define your boundary flows and concentrations. There are certain circumstances where if you place your boundary, it may not affect the simulation at all because it's far enough away from the problem. Usually this is in estuarine systems where you try to get your boundary closest to the ocean as far away from the problem as it can go, and whatever you put for concentrations there won't really affect the answer. So boundaries are kind of critical, and again they can be put in at any time scale in WASP. It can be every time step. They could be daily, monthly, annually. The information, all of the time series in WASP are, you can linearly interpolate them or step function interpolate them. So where can we get this? Where can we get this data? Well, for the flows, we can get flows from USGS gauges in our watershed, whether it's actual, or we can use a gauge and then calculate the flow at one of our boundaries based upon drainage area ratios. We can simulate using a watershed model that is driven by rainfall. You can set your flow conditions to a critical condition like 7Q10 or some other value. You can get flows for point sources from the NPDS databases. For water quality, you get concentrations. Again, you need to get concentrations for everything that's being simulated. You can use either the USGS NWIS database or US EPA STORA database to come up with quality if there are stations there. Some states have their own water quality databases that you can use, or you can go to an NPDS database. And just like you can simulate using a watershed model, you can use a watershed model to simulate the pollutant loadings as well as a function of rainfall, land use types, and other management activities. All right, the last set of input data that we're going to talk about is environmental. And this is basically environmental data that changes over time that affects water quality. This is things like solar radiation because the frequency and magnitude changes throughout the year. Cloud cover changes daily and it affects solar radiation. Wind speed, which affects reaeration and water temperature. Air temperature affects reaeration as well as water temperature calculations. Canopy shading affects both water temperature calculations and phytoplankton growth. If you're not using the sediment diagenesis model, you do have to provide some information about benthic fluxes, both for nitrogen, phosphorus, and sediment oxygen demand. And then lastly is zooplankton grazing on phytoplankton. So where do you kind of get this data? Well, most of this data is pretty readily available for the meteorological data, the National Weather Service. There's actually a download utility in basins that pulls down the NLDAS data, which is data that's been prepared by NASA using meteorological stations and radar data. Then you have your local W-band stations and a lot of different, like wastewater treatment plants may have their own meteorological stations that you could use. Canopy shading you can calculate using the inclination of the sun and the orientation of your network, and whether it's valley ridges or tree canopy, you can calculate it. Benthic fluxes, without the diagenesis model, you can either do field studies to calculate or to measure the values, or go to the literature to look at literature values. For zooplankton, you can either measure or estimate, and for a lot of cases we just ignore because it's a very hard one to get a grip on, and we kind of make up for the zooplankton grazing in the death rate that we apply to the model. The last set of data that you're going to have to compile is calibration data, and I would try to get any and all ambient monitoring data that's available. And keeping in mind that you have to make sure that the data is of quality for calibration purposes. But you want all the data that's available within the temporal and spatial domain of the model. And you're going to want to compare the model results to all of this data. If you don't, somebody else will. So I think it's very, very important to be honest with yourself and honest with everybody else and compare all of the data that's out there to the model predicted data. Alright, what we'll look at now is just a brief overview of the eutrophication model to see the things that are included in it and how they interact. For the eutrophication model, there's general water quality components that the model will simulate. That could be salinity, pH, solids, and water temperature. It can look at the impacts of nutrient enrichment. In the case of nutrients, it's nitrogen, phosphorus, and silica. It can look at impacts on dissolved oxygen, algal productivity, looking at 3 different types of algal species: phytoplankton, free-floating phytoplankton, periphyton, which is the algae that's attached to rocks, and then macroalgae, which is algae that actually could be floating on the surface of the water, floating in the middle of the water column, or down at the bottom. This is a schematic of the water quality processes that are considered in WASP. In a previous webinar, I presented the nutrient cycling in the aquatic environment. I would refer you to that webinar to see how all of this different information interacts and how it affects algal productivity. So the state variables for the eutrophication model are ammonia, both it's total ammonia, but it's broken out by ionized and unionized ammonia, and it will calculate unionized ammonia if you're simulating pH. Nitrate includes nitrite. Phosphate includes the 3 ionic forms of phosphate. Then you have dissolved silica, dissolved oxygen, salinity, alkalinity, total inorganic carbon, and then pH, and simulating up to 10 solids classes. For phytoplankton, you can have up to 5 classes. These classes could be specific assemblages, or they could be assemblages that are based upon light and temperature requirements. And then for periphyton macroalgae, you can have up to 3 classes of those, and those each have their own state variables for nitrogen and phosphorus because they use droop kinetics to take into account luxury consumption when they're growing. And then we have our particulate organic material, particulate organic carbon, nitrogen, phosphorus, and silica. And then we have our dissolved organic matter components, which one is carbonaceous biochemical oxygen demand. You have 5 classes of those. Dissolved organic nitrogen, and dissolved organic phosphorus, and silica. So the major processes are the nutrient cycling for nitrogen, phosphorus, and silicon, dissolved oxygen balance taking into account BOD organic material, and you can actually say sediment diagenesis. It simulates solar radiation, phytoplankton kinetics, periphyte and macroalgae kinetics, and then sediment diagenesis. So additional information that's needed by the eutrophication model is to adequately predict water temperature. You would need some meteorological conditions such like air temperature, wind speed, solar radiation, and the daylight that is fraction. In the case of phytoplankton, you also need to have solar radiation and daylight fraction. For water body conditions, you have to have water temperature, water velocity, salinity concentrations, and ice cover if you have it to affect dissolved oxygen. As we get ready to build our example application using WASP, I just want to take you through the steps in applying WASP. But these steps pretty much apply to all water quality models. The first thing is you need to clearly understand the questions the model will help inform a decision. There's nothing worse than selecting a model, developing an approach, and then find out later that they want a different kind of answer that the model was not parameterized, set up for, or it might not even be the most appropriate model. So make sure you understand what these questions are, what the management actions are going to be, and whether or not the model is capable of helping support those decisions. A big step is before you even begin the model is you should collect and review all data that's available that could be used in the modeling study, because the data can provide a good bit of information to you. It could give you the level and or cause of the water quality issue. You can identify prior to applying the model data insufficiencies, whether it's in developing the network, adequately defining the boundaries, point source, loading information, meteorological condition, or calibration data, because all of this data is very, very important to be able to support and prove that the model is adequately parameterized, and when it's calibrated there's enough data to constrain and show that the model is capable of helping in decisions. And then once you go through this data review, though it rarely happens with modelers, either develop or refine a monitoring plan to help address some of these insufficiencies if possible. But the first step certainly is you got to define the model domains. Spatially is what is the areas that you are going to want the model to simulate and cover, and then pick the periods of time that you want the model to simulate through, and hopefully these periods of time is where you have adequate data to parameterize the model, but it also includes the period of time that you have the water quality issue that you're trying to manage. So the model network, you can develop that, as I said, either NHD Plus or a WASP network tool, other methods, and if you're linking with a hydrodynamic model, it's already inherently done in the hydrodynamics. Define your flow paths to make sure that you're including all of the major tributaries that are coming into the model network, and provide the time series of flow. And you'll see in our example in just a little bit some of the ways that you can do that. Boundary conditions are very, very critical. As I said earlier, make sure you have time series of concentrations for every inflow and state variable. Make sure that you have environmental conditions that you can provide to the model to make sure that you are providing things that affect the kinetics in the model appropriately. And then you would begin the calibration process. And the calibration process is just that, it's a process. You may start the calibration process and realize that you need to go back and revisit some of your assumptions in the previous steps in setting up the model. So it is a process that you have to work through. So tips for modeling, and I can't emphasize this enough. I've been doing this for over 30 years. You need to keep your data organized. Document where all your data came from, who you got it from, if there's any data that you had to make calculations from, what data did you use to calculate it. Keep the data in a spreadsheet or a database. I prefer using something like WRDB because it was absolutely built to support water quality modeling. Because all of the data that you put into that tool, you can actually document where the data came from, so that when you look at it a year from now, you know what it is. The model development process is an iterative process, and you should check and verify at major steps in the development. The first thing is, one of the first checks you should do is once you build your model domain, your network, and your time domain, you should run the model using a conservative substance just to make sure that everything is connected appropriately. Keep multiple copies of major steps, because if you lose something, it could set you back a long way. As you are in the calibration process, keep a log of changes from one version of input to another. This way that you can always review back if something starts going the wrong way. It's always a good idea to have a second party take a look at what you're doing, just review. It doesn't have to be an in-depth thing, but peer review or by a coworker is always a good thing to do, because they may see something that you overlooked because you've been working so hard, the tree in the forest. And then document. It's critical that you develop a modeling report which documents the modeling approach, where all the data came from, your major assumptions. Provide calibration quantitative and qualitatively. And when I say that is that not only do you provide calibration statistics, but you should provide a series of time series plots that shows observed versus predicted, and you can do that several different ways. You should document the things that you did for sensitivity analysis and any major conclusions that were drawn from the modeling report. So, and why take the step of doing this modeling report and organizing your data? Well, you could be working on this project today, it comes to a halt, and then 4 years down the road they want to revive it and update the model. This provides you a good starting point for reviving the model and bringing it forward in time. During the development of this example WASP file that I'm going to show you, I'm going to be using a series of tools that I have developed, as well as the Water Resources database, which already has the data populated for building this file. At the end of the presentation, there'll be a screen with the links where you can download both the completed WASP input file, the Water Resources database that is going to be used to support this, as well as links to other tutorials that I've referenced during this webinar that would be helpful as you go forward using WASP. I just wanted to give you just some insight into some resources that are available from the WASP support website, which you actually can see inside the main window when you load the WASP model. First, this is where you would go to get the installation packages. We're about to release version 8.1, but you would come in here and you would select the version that you want to download. There's an FAQ for frequently asked questions. The first one here, is there an email support group? And yes, there is, with instructions on how to join it. We have workshop material for when people are coming to our workshops. But the two items I really wanted to point out to you is there's already webinars posted on how to do certain things on the website, and some of the ones that are important to you is I'm going to be showing you the linking LSPC with WASP, and it doesn't have to be LSPC, it could be HSPF, it really doesn't matter. I'm going to be showing you a little bit about using WRDB graph for model calibration. There'll be a brief demonstration towards the end of the webinar, and this is a better place to come and get some information. An introduction to WRDB, how to import data, and then there's one on how to use the WASP network tool, which I've already kind of demonstrated to you that would be useful. The other thing that's available online is the model documentation for the different components that you can just open up online or download, and it provides you with the documentations for the different modules. So that's what's available from the website, and whenever you launch WASP, you'll get this message here, and you will see if there's something that's updated or there's a new version available. It's going to tell you. What we're going to do now is we're actually going to create a WASP input data set using an example that I typically use for the WASP workshop. What we're going to do is it's going to be the eutrophication model. It's going to be simulated from 2006 through 2012. The LSPC watershed model is going to be used to provide daily loads and flows from the watershed to WASP. There's going to be 3 NPDS facilities in here. We're going to pull their data into the WASP model as well. I'll briefly show you a spreadsheet tool that's available for processing LSPC output to put it into a format that can be read in by WASP. We'll get all of the WASP meteorological conditions that we need will be extracted from the LSPC weather file, because that same data is required for the watershed model. All the data will be processed and imported into the water resources database, which WASP will open this database and read for input into the model. Just a little bit overview of the model that we're looking at. We already saw the segmentation that we've seen. We've got a main stem and 3 tributaries coming into the model and ultimately ending up down at the bottom in a lake. The watershed model, for each one of the subbasins, which are the red polygons, it will provide daily flows, total nitrogen, total phosphorus, BOD, DO, and water temperature to the WASP model. And each one of those subbasins will provide information to a specific WASP model. And it's just something you need to understand about watershed models. Output will be in 2 forms in a watershed model. One format is in the reaches of the watershed model, and the reaches are pretty much equivalent to like a channel. And so in the case of this subbasin here, 6103, we will be using the reach information, which would represent all of the flow and load coming out of this subbasin. For 6104, there is no pour point to the model, so we will not be using the reach information, because the reach information down here actually includes the flows and concentrations that were coming in from upstream. We will use the runoff component, which is basically the component, the contributions from the watershed to the side, and that's called Pero. So we have 2 different types of data we have to process out of LSPC. The pour point stuff, which is the RO part, the reach part, and then the Pero for the part that's not. So every subbasin here will be contributing to the WASP model network in one fashion or another. And it's critical as you go through this, you label which subbasins going into which WASP segment and what kind of linkage type is there, because it's critical to know that when you use the spreadsheet tool. This way you process the right data going forward to WASP. So in our case here, 6098 is an RO, 99 is a Pero, 100 is another Pero, 21 is an RO, and you can kind of see that if it's a headwater one, it's an RO. If it's one that comes in from the side, it's a Pero. So it's kind of critical to know this information. Okay, I've opened up the spreadsheet tool, the LSPC to WASP spreadsheet tool, just to give you a brief glimpse of what's in it. There is a tutorial on the WASP website on how to use this tool. Actually, it's for this specific example as well, so it'll take you all through it. But all I really want to show to you here is that you kind of have to be careful about lining up where the LSPC output is to what you want it to be processed on. And then WASP does not simulate total nitrogen, total phosphorus, so you have to fractionate the totals into the individual species. That's what these fractions are here. And I typically get these fractions by looking at the available ambient data to determine what it is. It's going to create 3 files that are listed up here. These files are what gets read in directly into WRDB to put the data into a format that WASP can use. And then lastly, there's the tab that basically points to where the LSPC files are, the type of linkage, so it knows which data to process, and then this information goes into the database so that we know what it is. The station ID is actually basically the LSPC subbasin number and the linkage type. And then we have the name, the latitude and longitude, so we could plot it in the GIS or not. And once you have everything lined up, you hit the button, it processes the data, and it will create these 3 files to import into WRDB. After processing the data from LSPC in the spreadsheet tool and processing and getting my point source data together and the meteorological data from LSPC, I imported the information into WRDB and actually created 3 working tables that we will later pull into to the WASP interface. The first one is the LSPC, so this is actually the LSPC output from the simulation that we're going to provide the flows and loads to the WASP model at the pour points. And it just has, this just gives you an idea of how much data is here. So for every state variable and for every station, there's 2,557 records, which is daily records for 6 years. So we'll be pulling that information in. The nice thing about WRDB is it gets the data all nice and organized and in a way that you can look at it. You can look at it in tabular format, or you could actually take it over and put it into a plot where you can plot the LSPC output. So in this case here, I'll just look at a flow at our headwaters. So we'll flow here. So that's our daily predicted flows. So this is a good tool for looking at that information. Another table that was created was the point source data, and the point source data has all of our data associated with the 3 treatment plants that we have coming into our model network. And again, it just gives you an idea of how much data we have, the min, the max, so on and so forth, and we can plot it. And the last chunk of data that was pulled in was actually the meteorological data that we imported from the LSPC meteorological file, which is called an air file. And again, it provides us with a breakdown of how much data is there. And in the case of the meteorological data, there's quite a bit of data here. There's 122,000 plus observations for each one of the parameters that we're bringing forward, because it's basically our data. So this database will be used to populate the WASP model in the coming steps. All right, what we're going to do now is I'm just going to take you through building a WASP input data set, and we're going to be using I'm going to be taking most of my information from a spreadsheet and all of my time series data from a water resources database, which I showed and reviewed with you just a little bit ago. So with everything organized, we have our WASP network is laid out here with all of the geometric information that we need for the channels, which was kind of derived from the NHD+. So we're going to load WASP. We're going to click on File. We're going to say New, which initializes the interface to everything's blank. We'll go ahead and we'll set the starting and ending time of the simulation, which is January 1st, 2006 at midnight, and we'll run through December 31st, 2012 at midnight. I'm going to just put a little description up here so it reminds me of what this is. It's just going to call it example watershed. And then the only other thing that I'll just call to your attention is by default the model type that's used is the advanced eutrophication, but if you're doing a toxicant, you would change it to toxicant model. Everything else we'll kind of leave alone. I don't want to get into too much of the details of some of the other specific stuff. It's all in the documentation. So the next thing we're going to do is we're going to add state variables to our model. Because WASP doesn't assume or know what you want to do, you need to add the state variables. So we're going to be adding the nitrogen series, the phosphorus series, DO, BOD. We're actually going to do 3 BOD components, and I'll explain to that in a little bit. We're going to add water temperature as well as solids, phytoplankton to simulate. So basically what this process does is you're telling the model these are the state variables that I want to consider, and the model interface will provide the input requirements for each one of those state variables that you select. If a state variable is not selected, you will not be exposed to any of the input data that's required for that state variable. So it kind of makes things a little bit more streamlined. So just finishing up picking up the state variables. So this is going to be solids, and the last one is going to be water temperature. So we'll actually simulate water temperature through the system. So the other nice thing is the interface allows you to label the state variables to something that maybe would relate directly to your data that you have, because you'll see how handy that is later on. So for the detrital, carbon, nitrogen, and phosphorus, this is the particular organic form of those. So that's POC, PON, POP. And then we have 3 BOD groups in here because they all will behave a little bit different. Biotic BOD is the BOD created from phytoplankton dying. The next BOD will be the BOD associated with the wastewater treatment plants, because they'll have their own decay rates. And then the last one would be the watershed BOD, the BOD that's washed off from the watershed model. And we're just putting more common names on the state variables here, just so when we start mapping in data from the external data sources, it's a little bit easier for us. So once we have that, we got 16 state variables that we're going to simulate. We'll click OK here, and then the next thing that we'll be doing is we'll be putting segment information in. The first thing is we're going to tell the model how many segments we have. So we click on the segment screen, and we're going to insert 34 rows. And then we will copy and paste the information from the spreadsheet into the interface. So the first thing we'll do is we'll copy the segment names in. It's kind of handy to have segment names with everything because, one, you'll see that it helps in mapping the external data into the model, but it also helps you know where certain things are in your network, like in this case, the wastewater treatment plants. The next thing is we're going to set the transport mode for each one of the segments. If you remember from the transport portion of the webinar early on, our lake is going to have a weir at the downstream end, and then all of the other segments will be, the water will be transported using the kinematic wave function. So basically this is just a drop-down list that lets you pick the flow routing option that you want to use. In our case, again, kinematic wave. So once we're done with that, we are done with this, so we'll click OK. And then the next thing that we're going to work on is actually putting the channel geometry in, which is basically going to be cutting and pasting from the spreadsheet. You'll see that the first column in here is our segment name, so everything lines up. So we're going to cut and paste the length and widths in to the interface. The model will calculate the volumes as a function of the lengths and widths. The kinematic wave, you'll see, needs slopes, and you'll notice that the ponded weir one does not. So if you're using a transport option that does not have required data, it'll be grayed out. And then we just set the minimum depth for the weir, or for the pond, to be, it doesn't go below 2 meters. And then we're just going to copy in the initial depths and the depth multiplier, which is again, we're going to start at the initial depth of. So this is like, these are all at average flow conditions. So once we're done with that, we've kind of set everything up. We're setting our weir height to 5 meters at the bottom, so when the depth of the lake at the bottom goes greater than 5 meters, water will go over that weir. We also could have not put a weir in there, but we actually could have put an internal flow boundary and had flow coming out. So the next thing we're going to do is we're going to define the flow paths. So we have 17 different flow paths. So this is where 17 different places in the model we have water and mass coming in, and we have to tell WASP where that's coming from and where it's going to. So we're going to use 17 flow functions, continuity functions to define that, and you can give them each names, and you'll see that basically we're going to start up at the main stem, which is the Ambrose River, and we basically tell WASP that the water comes in at segment 0 and it goes to 15, 15 to 14, 14 13 until it goes all the way out the bottom. So we're just going to paste that in. So we'll copy and paste the path, and it'll go into the interface. And we'll do this for the tributary. And you'll notice in this case it goes from 0 to 21 all the way down to 16, and then it just puts the water into the main stem of Ambrose River at segment 6. And you would do that for the other flows coming into Ambrose River. So this is Martin River, and it comes into Ambrose River at segment 5, and then the last one of the main tributaries is Flippin Creek, and it comes into Ambrose River at segment 3. So it's kind of critical to make sure that all this conductivity is correct. WASP will warn you if the conductivity is wrong when you try to run the model, but it won't try to correct it for you. The last things that we're putting in are the pero flows, the flows that come in from that come laterally in from the watershed model, and we're just putting them into the segment that the watershed subbation is associated with. So this is what the last sets of flow continuities are, and these are basically 0 to the segment. So for everywhere where we have a 0 to a segment, we're going to have to assign flows and concentrations of what's coming in, and we're going to get most of that from the watershed model. The ones for the wastewater treatment plants, we will be getting flows and concentrations from DMR data that we've processed. So that's pretty much how the flow screen works. If you look at it right now, you'll see that over in the lower right hand box, the grid that has the 2006 to 2012, that ultimately is where the time variable flows are going to come in. So for the watershed models, that would be the daily flows that would come in. For the point sources, that would be whatever flow data that we have for the point sources, and we'll look at that and review that a little bit later. So that's what the flow screen, that's how you define the flows. You define the flow paths where the water comes from and goes to, and then ultimately we'll be putting in a time series of flows. And it's always a good idea to go through and check this because you can make a mistake. And I'm just clicking through here, just looking. So once we do that, I'm going to save this. It's always a good idea as you build your input data set to save it as you go along. You never know, you may get a crash. So save as you go. All right, the next step we're going to do is we're actually going to link in our water resources database file to WASP. And the first one that we're going to start off with is we're going to start off with the LSPC, the watershed output, so our example watershed. And when you use this function, when you go to the database linkage, you got to make sure you use the same database engine type. In our case, it's SQLite. And I'm just going to browse to where the WRDB database is located. We'll open it up, and I'm going to go in there and I'm going to select the table that I pulled the LSPC in, which is the current. Because it is a WRDB table that we're going to, WASP kind of knows where everything is. So the first step we're going to do is we're going to map in the parameters. So this is basically we're going to map in the parameters from the database that we want to import to the state variables in WASP. So the state variables is the first column. You can see that the ones that we labeled, and then the parameter table is the parameter codes that are in the WRDB database. And we're just going to map in those parameter codes with the ones that are going to go into WASP. So we know that anything with DOP in the LSPC output will go into the organic phosphorus state variable in WASP. So we're going to add in the nitrogen series, the phosphorus series, DO, BOD, solids, and water temperature. That's all that's been predicted. The next thing we'll do is we will actually link the station IDs from the LSPC output to the WASP segments. And you'll notice here everywhere that there's a yellow, that is a boundary. And I was actually, everything was automatically mapped because I had my segment names set to the station ID in LSPC, so it's automatically mapped in. So that kind of lines up everything, so we really won't have much to do when it comes time to bring that information in. So the first thing we're going to do is we're going to link in the flows. So basically the way this works is we're going to use, we're going to click on the Ambrose River headwater. We're going to change the data source to example, the one that we just created. Set the P code to flow, and the flow here is in cubic meters per second, and then we're going to go pick the station, and the station is actually the segment name of 15 in this case, and then it's mapped in. And what WASP does is it actually goes to the WRDB database and it pulls that information in. What I'm doing here is I'm just quickly setting my data sources all to the same, the example, which is the LSPC output, and then I will set the P code to flow, and then I'll go set the station ID that's particular to each one of the inflows. So we've already got Ambrose's headwater, so all we have left to do are the other 16 flows that are here. So just you use the drop-down list. I'm going to look for 60101 RO, so on and so forth. And you'll notice as we're selecting these things, the graph on the right will update, showing the daily flows for each one of the subbasins that are being linked in. We will skip over the point sources right now because we did not link in the point source data. Remember that the point source data was actually imported into the water resources database as well. So we would continue this process down through all of the all 17 inflows to get the flows brought in. The other thing is not only do you get to see the graph, but if you look in the lower right table, you'll actually can see the daily flow values in the table there. So we'll let this continue on for a second as we as I load this information in, and then we can do a quick review of what's there. All right, that completes mapping in all of the LSPC flows. We're going to click OK. We're going to go back up, and now we're going to go ahead and add the point source data. We're just going to go to the external database icon. We're going to say our data source is going to be the nonpoint NPDS. We're going to browse to exactly the same database, but in this case here, we're not going to select the LSPC current table. We will select the NPDS table that we imported. And again, we'll go through the same process. We'll map the WASP state variables to the data that we have for the point sources. So I'm mapping in the nitrogen series right now. Then I'll map in the phosphorus series, and this is going to be all of the data from the point sources. Bring in dissolved oxygen, and then under BOD, I'm going to go to the wastewater treatment plant one and map in BOD. And then I think that's I think the only other thing we have is because we don't have solids is water temperature. So that'll map in the state variables and the P codes for the point sources. And then the next thing is we just need to go map in the wastewater treatment plant station IDs. That's Ambrose. And then we'll go down and we'll do Wool Industries and then Martin wastewater treatment plant. So that pretty much works the same way as it did before. And we'll go back over to the flows, and then we will just go into Ambrose and we'll change the data source to NPDS, and then we'll just pick on Ambrose, and that brings in the point source flow for Ambrose. And then we'll do the same thing for Wool Industries. You have to be a little tricky here because sometimes it jumps around based upon the letter that I was pressing, so that was kind of my mistake. So I got to go down and find Wool Industries. So now I'll pick the station ID here, and then the last one I'll do is Martin and change it to NPDS, and then pick the Martin station. So that brings all the flows in. So we've defined all of our inflows to our WASP network from LSPC and then from the point sources. And this is the boundary screen here. Because we've mapped in the parameter codes and the stations to the systems, we've brought in the daily concentrations that were predicted by the watershed model or the measured wastewater treatment plant automatically for each one of the state variables that we're simulating. So this is something that could take you a couple of days to cut and paste into WASP. It's brought in automatically from the database linkage. We're going to click OK, and then we're going to hit Save, and then we're going to add another one. And the last thing we're going to bring in, which can tend to be a tremendous amount of data, is meteorological data. Again, we're going to go back to the same water resources database file and then select the metadata table that was imported from the LSPC weather files. So we'll go to metadata, and we're not going to do we don't have any mapping to do here because the metadata has nothing to do with state variables. We're going to go to the time functions. So this is the things that can affect calculations at a vary in time. We're going to set the data source to metadata, set the parameter code to solar radiation, and then we're going to pick our station. In our case here, we really only have one station, so now it's going to pull in solar radiation for that from the WRDB table. We're going to do the same thing for air temperature. Select the data source, select the parameter code, air temperature, and then select the station. And by the way, the information that we're bringing in here is hourly, so it's changing every hour. So this is a lot of data that's being brought into the model, and it's being brought in, as you can see, very easily. And we'll finish up with we don't need cloud cover actually because the solar radiation was actually measured solar radiation, so it took into account light loss due to cloud cover. As we move on, it's just now we're just getting the dew point set, and most of the atmospheric data is used to calculate water temperature, except for solar radiation. That's used for water temperature and algal growth. So that pretty much takes care of all of our meteorological data. And just so you can see, that's 122,737 data points brought in for each one of those time functions. We're going to hit Save. Okay, once we've set those time functions, we need to assign those time functions to each individual segment. So because you can have multiple time functions for many of these things, different areas of your model network could be using a different time function. And we do this through the parameter screen. And virtually for every time function, there's two things on the parameter screen that you have to set. And these are segment specific. So I'm just going to go in here and I'm turning on temperature of the segment, which is a multiplier, or if you just wanted the temperature of the segment to be constant, you would set it to that temperature. And then for every one of the time functions, you'll have a multiplier and a time function number to point to. So in our case here, I'm basically setting the multipliers to 1, and then I'm telling the model that it should use the first time function entered for air temperature, wind speed, solar radiation, and dew point. If I had multiple time functions for each one of those time functions, like if I had 4 wind speeds, I would assign a different number, 1 to 4, for the time function to use, and that would allow different segments to use different time functions as it simulates through time. Okay, I just sped this process up fast in time just to save a little bit of time, but once I have everything set, I have the scale factor and the time function pointing to the right one. We're just going to click OK and save. Okay, we have 2 more steps in building this input file. The next one would be putting in the kinetic constants for changing to doing the fate and the phytoplankton growth in the model. There's not enough time in this webinar to go through all of these. There's model documentation that you can look at, as well as other webinars online. So what I'm going to do is I'm just going to show you how the constants are broken out. They're broken out by functional groups, and depending upon whether or not you have a specific state variable simulated, you will see them or you won't see them. So the first one is actually stuff for controlling the model time step optimization routine. Usually you don't have to touch any of this. And then they're broken up by functional groups: global constants, constants related to predicting water temperature, inorganic nutrient kinetics. These would be the rates. If you have partitioning of phosphorus or ammonia to solids, this is where you would set the partitioning coefficients. Organic nutrients is where you set the mineralization rates for the various organic components. BOD is where you would set rates specific to the multiple BOD groups. The same thing is true for dissolved oxygen. Phytoplankton, if you're doing more than one phytoplankton group, you would have all of that in there. Setting information for the light field as far as light extinction. If you were doing macroalgae information. Constants and kinetics for the sediment diagenesis model, constants and kinetics for the solids transport, that's the mechanistic transport, and then bacteria. So the way these kind of work is, I'm only going to take you into one, but if I go into the inorganic nutrients, you will see that you have a description of what the constant is, which system it's going to affect. You'll actually have a value here that you could use. And you'll have a box that you check. When it's checked, it means it's sent to the model. If it's unchecked, it means it's going to use whatever the default is here. So you check it, and that information gets sent to the model. So basically, here's your nitrification rate, the temperature correction, and it goes all the way on through phytoplankton, where you have all of the different constants that you would use. And as I said earlier, this input file will be available for download, so you can look at some of the constants that I use. So that's where you set the kinetic constants. The last thing that we're going to need to do is we're going to turn on some output, because output is not turned on automatically because it doesn't know which state variables you're using. So if it's checked, it's going to get outputted. So we don't output everything. You get to pick and choose, and again, it goes by functional group. You pick what you want to output. And there's also another file, output file that WASP creates, and that's a flux file. And basically this will output all of the different fluxes pertaining to a different state variable. So I'm just going to turn on, I'm going to ask for the amount of oxygen lost or gained from reaeration, from phytoplankton productivity, DL loss to nitrification, BOD decay, phytoplankton respiration, and so we'll have an output, and we'll look at that. So with that, I'm going to say OK, save, and then to run the model, you click on the WASP, and the first thing the model is going to do is it's actually going to query the database to bring in all of the data from the WRDB tables. And this is very important, because why use the database? Well, one, it keeps you from typing it in. Two, if you had a different scenario that you wanted to run and you did some modifications to the point sources or to the LSPC, all you'd have to do is point to another table. So once the data is read in, the model will take off and run. This grid up at the top just shows you the calculations that are being made at the current time. So you get to see most of the state variables. You get to see some environmental fields like light, temperature, and then out here you'll have the transport, water velocity, depth, so on and so forth. And you can see it's all changing in time, and it's moving right along. If the model encounters any issues that one, it can't fix, or it's going to fix, it's just going to issue these warnings, or if there's an error, it'll tell you what the error is. So I'm just going to let this continue to run, and we'll rejoin. Once it's done, we'll talk some more. All right, as the model's about to finish up, I'm going to take you through how you can look at the model results using the post processor, which utilizes the Water Resources Database Graphing Program. So the model's just finished. It took a little bit over almost 4 minutes to run those 6 years. So what we're going to do is we're going to launch the post-processor to look at some of the results. There's 2 options here. One of them will launch the BMD utility program that comes with WRDB. The other one will be the graphing program. So I'm just going to launch the graphing program. It loads in our data set, and the way the graphing program works, it's broken up into 2 components. You have your station information, or in our case segments, and you can see that our segment names were carried over here. So we're mainly going to look at Lake Sarah, and then these are the output variables that we have available to us that we can plot. You do have the ability to open more than one BMD file at a time. So like if you were comparing a base case to a reduction case, you can bring them both in here and plot them against one another. So the kind of plots that you have available in here are, well, one, a conventional time series plot. So I'm going to go ahead and I'm going to plot total phytoplankton for Lake Sarah, and we get a plot that looks kind of like this. This is the data that was there, that was produced by the model. You do have the ability to change and modify anything the way it looks. When you load the WRDB graph, the default doesn't look like that. You can go under the options tab here and just change just everything the way it looks. It's under this tab here, and mine's pretty much geared for publications. You also can plot multiple time series at a time. So in this case here, I'm going to go and get all 3 of my phytoplankton species that I'm simulating here. I'm going to add them to the left axis, and just for consistency's sake, I want them all to be on the same axis so that the units stay the same. So this is kind of where we have the blue is our fall bloom, the black is our spring bloom, and the red is our summer bloom. So we can look at things that way. Another thing that we can do is we can actually do a probability plot. So if I wanted to look at, let's say, dissolved oxygen in Lake Sarah, I can get a plot, and we can see that, pretty much 50% of the time it's up around 9.4 milligrams per liter. This is a good way to how you can do comparisons of frequency of exceedances. You also have the ability to do longitudinal profiles, and we'll do this by river mile. So what we'll pick here is we will pick well, let's pick total nitrogen, because I know there's a big signal of total nitrogen in the system, and we add that to the system. So now keep in mind that the profiles are going to be at a specific time. So there's a slider down here that lets you pick. Now each one of these are actually a branch. One is Ambrose River, the other one is Martin, Wool, and Flipping Creek. If you wanted to just do one branch, you can change it to pick. We'll do the first branch, which is Ambrose Creek. So you can do longitudinal profiles. You also can add plots. So I could put water temperature on the left, dissolved oxygen on the right, so you can see. Oh, I grabbed the wrong thing. I grabbed reaeration. I'm going to just change that to dissolved oxygen, and you can see, as you should get, the inverse relationship between the two. So there's plenty of things that you can do here. You also can open up, and there's a more sophisticated way, but I'm going to open up the watershed WRDB, and I'm going to bring in the observed data. So you also, from here, I can go ahead and, let me just remove these. So I can go ahead and go Lake Sarah, I can go total nitrogen plot. So there's the observed data, and then I can go back and say, okay, well, let me go get Lake Sarah total nitrogen to plot the two, and lo and behold, we're fairly well calibrated. So once you get a bunch of graphs the way that you want, you can actually save this layout to something with a .graph file, and I'll just call this watershed. Come on. And it'll save it as a graph file. So now we can go back over to WASP, and in the project we can remove the BMD, and we can get that .graph file. And then every time you run the model, you can pull, the graphs will be automatically created. So in this case, if I save it here, I could make another run, and if I hit this button, all the graphs that I built are there. So there's my TNTP calibration plot. So it's very, very handy. The last thing I wanted to show you is I was going to open up that flux file just so you can see how that works. Just got to get to where it is. So now in the flux file, what we have is, well, we only turned on the DO once, but basically for every segment we can get a DO budget for what's happening there over time. So let me remove these from here, and I'm going to pick Lake Sarah and all the DO. And again, you're probably going to want these all on the same scale, so it's all relative. But there you can see the red is phytoplankton production. The black is the gain. Actually, it's the loss to reaeration. So there's periods of time that the lake is supersaturated and it goes out. The blue line here is respiration. The smaller navy blue is nitrification. So it gives you a good idea of what's going on in your system. So this is the WDB graph is a great tool for post-processing data. All right, I'm just going to give you a quick overview of the hydrodynamic linkage. Building the input data set is pretty much the same whether you're using a hydrodynamic linkage file or not. The only real big difference is with the hydrodynamic linkage file, the model network is derived from the hydrodynamic model itself. So pretty much any spatial component of the hydrodynamic model would be provided to WASP. And what the hydrodynamic model does is it provides the segment volumes, the segment water velocities, segment depth, water temperature, and salinity, and the flows between all of the cells. If it's a multidimensional model in all directions, as WASP iterates through time, it reads information from this file and updates. So in my little example here, we're just going to take this is a very simple grid for an EFDC model for a small lake in Florida. And I'm just going to show you that this is actually the model domain that was generated for EFDC, and you will see that WASP will use the same domain when we pull the hydrodynamic linkage file. All right, so I've loaded WASP. I'm going to say I'm going to create a new file. I'm going to go to the dataset parameterization screen. This is going to be an advanced eutrophication model. I'm not going to do anything with this, the date and time regime, because all of that is controlled by the hydrodynamic file. So I'm just going to click hydrodynamics, and then I'm going to browse until I get to the hydrodynamic file that I want. I'm going to select it. It's going to come here in here, and I'm just going to say OK. And what happens is WASP opens up the hydrodynamic linkage file and it reads information in. So in our grid there was 425 cells, which turns out to be segments. In those 425 segments, there's 822 different flow paths, so flows going in all directions. The hydrodynamic model time step is 60 seconds. The WASP time step could be anywhere between 60 seconds and 30 times 60 seconds. WASP will optimize its time step to make things quicker. The start time of the model is January 1st, 2006, and it ends on 12/24/2012. And it will simulate from there. So what I'm going to do, I just want to go in here and I want to just change my output interval. So instead of coming out almost every hour, I'm going to have it come out 4 times a day. I'm going to click OK. I'm going to add a state variable because, as I showed previously, it doesn't assume anything. I'm just going to add ammonia. I'm not going to add any kinetics. And then I'm just going to go ahead and save this. And when I click run, it's just going to take off and run. And what you can see, the time is clicking by down here. And you will see, because this was a hydrodynamic linkage file, one of the requirements of the hydrodynamic model is it needs to predict temperature. So the temperatures are coming in, the velocities are changing, the depths are changing, the volumes and flows are changing. So all of that information is being read from the hydrodynamic file. You would still be responsible for putting in the environmental time functions, the segment parameters, the constants, and you still have the ability to link to external databases for your time series, for your boundaries, because now you'll have a lot more boundaries with a hydrodynamic linkage file. So it kind of works all the same way. All right, I'm going to give you a brief overview of the calibration process that I use. There's many different ways to do it, but one of the things that we have to be careful about when you start calibrating something like a eutrophication model, there are so many things that can change, and you kind of want to do it in a stepwise process. So basically, my calibration process goes something like this. Transport, you got to make sure that you have the flows and travel times correct. Because if these are not correct, anything that you do later to calibrate the model, you're trying to overcome not getting the transport correct. It's critical to make sure that this is best represented in the model. When it comes to the initial calibration, I do an initial nutrient calibration with no kinetics turned on, but I have all of my boundaries set. I run the model and I plot total nitrogen and total phosphorus predicted by the model versus the observed. If the model is under-predicting the observed data at this point, you're missing loads. There's nothing that you can do to raise total nitrogen or total phosphorus. The model does not create mass. If you're over-predicting, it might be okay because you have things like assimilative processes, settling processes that would lower the amount in the water column. But if you're over by a large factor, you may have to go back and revisit the loading assumptions. The first thing I do is I do a rough calibration between dissolved oxygen and BOD, and that basically means make sure that the DO-BOD curves are kind of representing where the data is. Maybe not so much in value, but in shape and seasonality. If you have a system that has a lot of phytoplankton in it, and it's not a riverine system, in all likelihood you will be under-predicting BOD by a little bit. So you know that if you're under-predicting BOD and over-predicting DO, you know that you'll have some biotic-generated BOD once you start the phytoplankton. Then I do a piecewise, I start to do a rough calibration for nitrogen. The first thing I do is I look at the ratio of ammonia to nitrate. I'll adjust my nitrification rate to kind of get that in the ballpark. And then I'll also look at my organic nitrogen to ammonia to adjust that mineralization rate so they kind of get in a balance. So in this case here, that step is only involving two rate constants that can get you into the ballpark. And it's not that you're going to be done with them, say, but it's a good starting point. And then I do the same thing with the organic phosphorus to the inorganic phosphorus, just looking at the mineralization rate. And then once you turn on the phytoplankton and start parameterizing that brings into the equation a lot more rate constants that have to be looked at. But things that you're going to want to make sure that are correct are you're going to want to make sure that your light field is correct, your amount of inorganic nutrients is close to what's been measured out there, and then you would begin the process of adjusting the growth rates, the death rates, the respiration rates to calibrate the phytoplankton in. And it is a iterative process. You're going to learn as you go. I can't give an absolute step. If you do this and that, at the end you'll end up with a calibrated model. It takes an iterative process, hundreds of runs. Before we stop and take questions, I just want to introduce you to the WASP development team and just give you a little bit of insight of where we come from. One of the first things is how has WASP survived? WASP is one of the models that was developed and supported by EPA in the 80s and 90s. How did it stay alive today without any dedicated funding, any commitment by EPA to keep it alive? It survived because of it had champions, people who believed in this model, who were willing to put extra time and effort into making sure the model was kept up to date and had the state-of-the-art science in it. And a lot of the things that were added to the model was added because we needed to do our job. We needed the information or the methods in the model to provide answers to regulatory questions, mainly for the TMDL program. It's one of the reasons why the sediment diagenesis model was added, is because we needed to be able to account for changes in sediment oxygen demand as changes in loads. So let me introduce you to the WASP development team. All of us have worked together for at least 25 to 30 plus years. Alex Comer has been responsible for development of the user interface and the algorithms used for the efficient post-processing. I've been the orchestrator and I guess the conductor of WASP development since probably 1995, working with Bob Ambrose and James Martin to work on the scientific code. Bob, of course, is the father of WASP, brought it to EPA. Bob early on saw the utility of WASP and how the generalized framework could be used to do different water quality constituents. And I think it's Bob's drive that brought the other three of us together to work on this code. James Martin, who is a professor of engineering at Mississippi State, had early on in his career saw the utility of WASP and actually changed his career paths to become involved with WASP. So that's our team. We've been together for a very, very long time. And again, this is something very, very unique in the water quality modeling field.